All right, good afternoon. My name is Hunter, and this is content review for Principles of Biology 211. Today we're looking at Mendelian genetics. Um, I am going to apologize in advance. Losing my voice a little bit. I might stop here to drink water a little more often and mute to cough here and there. Uh, but I think we're going to be all right, and we're going to get through it. Um, okay, first, a quick vocabulary overview. I really like biology for lots of reasons, but one of them is just because of like how precise the language is and um, how many ways there are to say the same thing can be kind of disorienting, especially in a topic as complex as genetics can be. But I think really getting a grasp of the vocabulary here can make a lot of the problems a lot more approachable. And so spending some time here and making sure we really understand these terms is absolutely worthwhile. So we're going to start with a simple matching exercise. Match these commonly used genetics terms with its appropriate definition or example. First one here is heterozygous. Prefix hetero, meaning more than one. Zygous, this should be familiar from the exercise we did last week with mitosis and meiosis, zygotes being important for reproduction. So we're looking specifically at specific genes and chromosomes and traits. So heterozygous is going to be this one right here. We've got a big B, little b, and this is just how we represent um, different alleles between, or yes, different alleles, different characteristics in an individual. And then, of course, we've got homozygous, so that could be big B, or little b, little b. Then we have a monohybrid cross, prefix mono, hybrid meaning, I guess, a combination. Monohybrid cross. This one would be a monohybrid cross. I, the answer here is G, a blonde mating with a brunette, as the example. Blonde, I think we're assuming, is homozygous recessive, little b, and a brunette could be big B, or big B, little b, if we're assuming that brunette is uh, the dominant trait. And I guess the way that these are capitalized, this may actually be a hint about which one they're saying is dominant. Blonde in this case would be dominant, but I digress. Autosomal. So um, in biology, anytime you see soma, uh, somatics, somatosensory, and this is to do with the body, auto, self. So this term really just means anything not on a sex chromosome. Um, but that is the origin of that term. So um, genotype. Ah, I like this. So your genotype is going to be exactly the alleles we've been describing: big B with little b, or little b with little b. This is H. Genotype is the gene that exists, not necessarily the trait that is expressed. Right? That would be phenotype. Phenotype is what gets expressed. So blue or brown eyes is our example in this one. So let's go ahead and let's see what we've gotten through. We've gotten E, B, G, C, through H, and D. A gene. Gene is genetic material, the genetic information, locus on a chromosome that codes for a given polypeptide. Remember the central dogma of biology is um, DNA translated into RNA, transcribed into protein. Um, the gene is what codes for that. Um, then we have an allele. Um, the answer here is I. Males have only one for each gene on the X chromosome. There's only one allele for each gene on the X chromosome. And then we have our dihybrid cross. Our dihybrid cross, of course, then is going to be A. Yes, we already used that. It's going to be A. So a blue-eyed blonde mates with a brown-eyed brunette dihybrid cross. That's going to be, well, we'll get to that in a moment. Okay, to become fluent with these terms, we're going to get more practice with them as we go through the worksheet. So from a single sex cell going through meiosis, how many daughter cells are produced? We went over this last week. Remember that meiosis or meiosis, whichever pronunciation helps you remember that it's different. Meiosis produces, there's two divisions. You have meiosis 1 and meiosis 2. And so from 1, you get 4. 4 cells produced by the end of meiosis. For your model organism or individual defined in question 1, how many different kinds of gametes can be produced from a single cell undergoing meiosis and assume no crossing over occurs? For your model organism or individual defined in question 1, this is from a single sex cell going through meiosis, how many daughter cells? So a single cell from this individual that undergoes meiosis with no crossing over, remember that's that conjugation where we get a lot of genetic diversity. We're simplifying this just a little bit. This is kind of the magic of intro Mendelian genetics. A single cell from this individual that undergoes meiosis with no crossing over is going to produce two different kinds of gametes. Two different kinds of gametes. And we're cut off here, but that's okay. Yeah. Your individual is heterozygous for 2 genes on separate pairs of homologous chromosomes. Their genotype is big C, big B, little b. Given this information alone, how many different kinds of gametes could this individual produce? And again, assume no crossing over occurs. So heterozygous individuals, okay. You could, on average, produce 4 different kinds of gametes. Hello, welcome. And they're all going to be in equal proportions. We can build a table to kind of demonstrate this. But basically, we can end up with big C, big B, big C, little b, big B, little c, little b, in equal proportions. Excuse me. Excuse me. All right. Now, compare your answers to the previous 2 questions. How do the numbers of different kinds of gametes in your answers compare, and explain any difference? So any single cell, right? Single cell going through meiosis with no crossing over produces only 2 types of gametes at most. However, that individual has the potential to produce four different types of gametes. The particular combinations of C and B alleles in the gametes is a result of how those chromosomes line up in metaphase 1. That's where that conjugation happens. So this could line up at metaphase 1 in a couple of ways. And we're gonna make a little chart here, but that was a lot of talking, and I want to write this down so that we can make sure we get all this. So single cell in meiosis. We're going to run out of room, but that's okay. Produces 2 max kinds of gametes. Um, that individual can produce four, can produce four kinds of gametes. We're going to make a little chart here. And let me think how I want to do this. Yeah, we'll just do it quick and dirty. It'll be messy. So, individuals A and B. We've got, how do I want to do this? So, Trisha did a really awesome job of color coding all of these things. And I think that is really helpful to kind of track where um, things come from and where they go when you're working on these tables. I think I'm just going to have to go for it right now. We're dealing with C's and B's. So, all right, and C, B, C. Okay, let me, hang on 1 second. Now, I just wanted to make sure that I was understanding the question properly and that I was explaining it properly. Uh, Basically, when we how do the numbers of different kinds of gametes in your answers compare? Um, yeah, when we have these heterozygous individuals with two different traits that we're looking at, um, then whoop, meant to use my highlighter here. Um, we have opportunities for all of these various combinations, um, depending on which one is conjugated in at the metaphysical plate where conjugation occurs in meiosis one. So yeah, let's move on. So what aspects of meiosis account for Mendel's law of segregation? So Mendel's law of segregation. Let's remember for a second. Um, this one is the one that states that each organism contains two traits. Today we call these alleles for a given character. Character we would now call it a gene. Only one allele is found in each gamete. Synapsis of homologous chromosomes in prophase 1 and their separation to opposite poles in anaphase 1 separate or segregate alleles of a given gene into different gametes. So Mendel's law of segregation um, basically describes the behavior of separate genes. So how do I want to? Each organism contains two alleles for a given trait, but only one is found in each gamete. Um, Separation occurs in prophase one. I'm trying to think what's the most important thing to have like include here. Um, yeah, synapsis during prophase one and separation in anaphase one segregate alleles. Between gametes. Cool. So Mendel's law of segregation has to do with gene behavior, and Mendel's law of independent assortment has to do with chromosome behavior. So Mendel's law of independent assortment states that the pairs of traits that control each character act independent of each other in gamete formation. So today we know that the assumption is that these traits or alleles are on separate pairs of homologous chromosomes. In other words, how one set of traits on one pair of homologous chromosomes segregates in gametes does not affect how another set of traits on a different pair of homologous chromosomes segregates. Let's write that down. So uh, let's see. The amino yeah. Pairs of traits, or yes, pairs of alleles. We're going to say alleles. That control each trait, excuse me, each, yes, trait. Got to stay on top of the language here. Um, act independent of each other on gamete formation. Act independent of each other on gamete formation. Yeah. Okay. All right. Now we've got this really helpful guide. This is a fantastic step-by-step where you can fall back on this. I would include some version of it in your study guide as you're doing practice problems, and practice is really the way to go for preparing for genetics-based questions, and these will be on your exams in Dr. Reddy's class. I know for sure. So over the, over the years, rules have been developed for setting up genetics problems and denoting genes and their alleles in these problems. And this activity provides a quick review of some of these rules. After you've read through all of this material, complete activities 14.4, 15.1, and 15.2, highly recommend. Do it. Basic assumptions to make when solving genetics problems. Are the genes linked? If the problem does not A, indicate that the genes are linked, or B, ask whether the genes are or could be linked, then you should assume that the genes are not linked. Are the genes sex-linked? Similarly, if the problem does not A, indicate that the genes are sex-linked, that is, on the X chromosome, or B, ask whether the genes are or could be on the X chromosome or Y chromosome, then you should assume that the genes are on autosomes and are not sex-linked. Is there a lethal allele? And it looks like we got cut off here, so it looks like we should be able to read it all. If a gene is lethal, lethal being the operative word here, then you should assume that the offspring that get the lethal allele, if dominant, or alleles if homozygous recessive, do not appear. That is, they are not born, they don't hatch, and so forth. Um, Therefore, you are not counting, they should not be counted among offspring. So an obvious exception would be lethal genes that have their effect later in life. And if this is the case, it should be noted in the question. So do not appear. Do not appear in the population. Are the alleles dominant, recessive, or neither? And unless the problem states otherwise, assume that capital letters, big B, for example, designate dominant alleles, and lowercase letters, little b, for example, indicate recessive alleles. When there is codominance or incomplete dominance, it's important to know the distinction between those. The alleles are usually designated by the same capital letter, and each one is given a superscript. For example, C superscript R, C superscript W, in figure 14.10 of Campbell Biology 11th edition. You will see, I guess, more examples of that. Great. So there's your notation. And I'm going to go highlight in green here codominance or incomplete dominance. Know what those mean for your exam. How are genotypes written? So assume a gene for fur color in hamsters is located on the number 1 pair of homologous autosomes. So autosome, not sex-linked. Brown fur, big B, is dominant over white fur, little b. The genotype for fur color can be designated in different ways. A, the alleles can be shown associated with the number 1 chromosome. In this notation, an individual heterozygous for this gene is designated as big B, little b. But most commonly, this notation is simplified to big B, little b, as shown here. I have never seen this notation except on this worksheet. Not to say that you shouldn't know what it looks like. In problems that involve sex-linked genes, the chromosomes are always indicated. For example, X big A, X little a, and X little a, Y. So in this case, if you're an individual with XY chromosomes and a recessive gene shows up, it will be expressed. So these are important things to remember about. There are clues in the language that can allow you to draw conclusions about what will be expressed. It's a little language puzzle. I love it. All right. What information do you need to gather before trying to solve a genetics problem? Before trying to solve any genetics problem, answer these questions. What information is provided? What type of cross is it? Is it a monohybrid, a dihybrid cross? Are the genes sex-linked or autosomal? Are they linked or unlinked? What does the information, I'm gonna just put a big old, know this. What does the information provided tell you about the genes in question? For example, what phenotypes can result? Phenotype being what is expressed versus genotype, what alleles are present. How many alleles does the gene have? Are the alleles of the gene dominant, recessive, codominant, etc.? Does the question supply any information about the individual's genotypes? And if so, what information is provided? Grandparent information, parental information, gamete possibilities, offspring possibilities. You will be able to kind of reverse engineer what the genotypes might be based on what gets expressed by offspring. And that will come with some pattern recognition and just doing a lot of Punnett squares. I highly recommend doing the Punnett squares and multi-generational Punnett squares. So what is a Punnett square? Punnett squares are frequently used in solving genetics problems. A Punnett square is a device that allows you to determine all the possible paired combinations of two sets of characteristics. For example, if you wanted to determine all the possible combinations of red, blue, and green and shirts with red, blue, and green pants, you could set up this Punnett square. So shirts up top, pants on the left. Red shirts with red pants produces red shirts and red pants. Blue shirts with red pants produces blue shirt and red pants. And it's occurring to me that I should really be using highlighters because red pants, red shirt, blue shirt, red pants, green shirt, and red pants. You can do this with anything. Now, I'm going to go ahead and change this because the red shirt gene allele from one parent came from one parent, and the other red shirt or red pants, yeah, they're different. It's not color coding, not based on what gets expressed, but based on what the gene is important. So I wanted to go back and correct myself here. Red shirt gets yellow highlighter. Red pants gets the red highlighter. Cool. And so on. Right? Let's do, well, the color coding is helpful for tracking where things came from. It is a good practice. It's not the easiest thing in the world if you're doing this with pen and paper. It's why I really like these digital note-taking systems because you can go back and correct it pretty easily. But it's a good practice to be in. Okay. So similarly, if you wanted to determine the probability of a male XY and a female XX chromosome having a son or a daughter, you would first determine the possible gametes each could produce, and then set up a Punnett square to look at all the possible combinations of male and female gametes. So here, meiosis dictates that the female's gametes get one of her X chromosomes or the other. You have two X chromosomes, but it's going to be one or the other. In the male, the gametes get either the X chromosome or the Y, and as a result, the Punnett square would look something like this. So you have female gametes X and Y, male gametes X and Y. I meant XX, sorry, XX and XY. And so we've got the female X here and the male X here. Male Y. Stayed in their respective columns and rows, combined to make something novel, something new. Okay. Now that we've seen that done a couple ways, let's get into some problems. So if you know the parents' genotypes, how can you determine what types of offspring they will produce? For autosomal genes, an autosomal gene that has the alleles big A and little a, there are three possible genotypes: big A, big A, big A, little a, and little a, little a. And this is the same as the X and Y exercise we just did, but sorry, I'm going to keep reading and then we'll do the Punnett square. All possible combinations of matings and offspring of two individuals carrying the autosomal genes with alleles big A and little a are shown in the figure below. If you know how to solve these 6 crosses, you can solve any problem involving one or more autosomal genes. This right here is why you should do these exercises. It is worth it. All right. It's worth taking a minute to go through these. So if we pair a homozygous dominant individual with another homozygous dominant individual, 100% of the offspring are going to be homozygous dominant for that big A gene, or allele, I guess, right? If we have a homozygous dominant with a homozygous recessive, right, 100% of your offspring will be heterozygous for those alleles. 100% of them. This is where that pattern recognition really comes in. If you know that your individuals are homozygous, the same or different, you have 100% certainty of what they are going to be. The difficulty comes when you have something like this, where you have a homozygous dominant or recessive individual with a heterozygous individual. The heterozygote, I'm going to do it this way. How do I want to do this? Yeah, yes. Yeah, I want to do it this way. Okay, so we have our homozygote over here, heterozygote up top. You can just carry these down to kind of simplify, but in this example, only 50% end up with that recessive little a gene, but 100% of them express the dominant gene. 100% of these express the big A phenotype, whatever that is. And by contrast, we're going to do one more just as like good practice. Here we have a homozygous recessive with a heterozygous individual. And here we get 50%. 50% express the dominant gene, and 50% express the recessive gene, or homozygous recessive, because the homozygous recessive individual is the only one that is going to express that recessive trait. Okay, note, if you take sex into account, there are actually 9 possible combinations of matings. This is where it starts to get really interesting. Because the results of reciprocal autosomal matings, for example, big A male with a little a female, and little a male with a big A female, there are the same, only one of each reciprocal type is included in the 6 combinations above. Right? Sex-linked genes that have 2 alleles, females have 3 possible phenotypes, and males have only 2 possible phenotypes. All the possible combinations of matings and offspring for a sex-linked trait are listed in the next figure. If you know how to solve these, you can solve any genetics problem involving sex-linked traits. Let's look at the one that I know will probably, well, okay, so they have a superscript W plus, I guess, but I'm not sure. Yeah, I'm not going to spend too much time looking at that because I'm not 100% sure what they want to ask on that. Looking at multiple genes, remember if genes are on separate chromosomes, then they assort independently in meiosis. Therefore, to solve a genetics problem involving multiple genes, where each gene is on a separate pair of homologous chromosomes, you have to solve for each gene separately and determine probabilities for combination, multi-gene genotypes, by multiplying the probabilities of the individual phenotypes. And just a little review of probabilities, I guess. If there's a 50% chance probability for one trait and a 50% probability of another trait, the chance that an individual has both of those is 25%. Same thing written a different way. Um, there was a fun thing on social media not long ago of like, what are the odds that you get three left-handed people in the room at the same time? If it's sampling the general population, and it was something like 1 in 10 people is left-handed. Um, and so, to get two people who are both left-handed in a room by chance, it was something like it's 1 in 100, and to get three people, it's times 10 again, so one in 1,000 kind of thing. Um, Simple way to kind of visualize that. We're doing the same kind of probability math here. um, yeah, so you multiply the probabilities together. Okay, where were we? What is the probability that two individuals of the genotype big A, big B, and a, big B, will have any a, offspring? To answer this, you have to solve for each gene separately. Let me give you a good example here. So we're trying to solve for any a, b. So we've got a is 50%, and we've got b is 1 in 4. So 1/2 times 1 in 4 gives you 1 in 8. Is that right? Yes. Your probability is 1 in 8. And you can figure all of this out just by literally counting boxes in Punnett squares. You do not have to be particularly good at math to be able to do this. It gets a little more complicated as we get into greater and greater numbers of genes and alleles that we're trying to do here. All right, how can you determine all the possible types of gametes? To solve genetics problems in which genotypes are given, you must first know what types of gametes each organism can produce. So how many different kinds of gametes can individuals with each of the following genotypes produce? And we're given a homozygous dominant individual. And this is n equals 1. One kind of gamete. Different color here. One type of gamete. My handwriting is a little bigger than this type, so we're going to, yeah, there we go. And that is big A. Same for the homozygous recessive. For a heterozygote, big A, we've got 2. And we can have A, big A, or little a in equal proportion. All right. Now we're adding an additional gene. So big A, big B. Here we can have 2 kinds of gametes, also in equal proportion. We can have big A, big B, or little a, big B in equal proportions. Make sense? Only 2 options. All right. Big A, big B, little b. based on the exercise we did above, you should get four. Four kinds of gametes. You can have big A, big A, little a, big B, and little a, little b. And these would also be in equal proportion. Okay, adding an additional gene. It's homozygous dominant. Okay, we can shortcut this. This would basically be like multiplying times one, right? Um, all offspring are going to have something from column A, something from column B, and something from column C. If all of the column C ones are the same, that means that we just have to worry about the differences between A and B, which means we're going to have four. And that's it. Now, that breaks down when we have a third heterozygous trait. And here, you would get 8. And we are going to write all of these out. A, B, C, A, B, C. A, B, C. And brain broken, come back. And we've got 2, 3, 4, 5, 6, 7. I'm forgetting one. A, B, C. There. Cool. Now, I'm not going to write all of these out because we would run out of room and it's going to take a long time. But we are, and I'm just going to alternate here just to make it easier for my eyeballs. All of these are heterozygous, which means that each of them is going to contribute the same amount of potential complexity, right? We have 1, 2, 3, 4, 5, 6 different allele pairs now that we're looking at. And if 3 of those produced 4 different traits, you can bet. Is that how I want to explain this? Yeah, excuse me. 3 of those produced 8 different pairs. If we're simply multiplying by 2, that wouldn't be right though. It'd be 5 times 4. Yeah, sorry. I'm tired, y'all. um, yeah, no, it's sorry. It's not times 2 even. It's times 4. You would end up with 32 different kinds of gametes all in equal proportions. That doesn't, hang on. Something doesn't feel right. Check our work. Times 6. Yeah, I don't think. Yeah. Yeah, this is wrong. This would be 1, 2, 3, 4, 5, 6. They're all heterozygous. This would be 64. Yeah, this would be 64 different kinds of gametes, all in equal proportions. And we know this because of this very next prompt. We can come up with a formula, and this will be very helpful. Based on our answers in question 1, propose a general rule for determining the number of different gametes organisms like those described in question 1 can produce. And it's simply the number of gamete types equals 2 to the power of n, where n equals the number of heterozygous alleles. And here the assumption is that the different genes are on separate pairs of homologous chromosomes. And so for this example H, we had 1, 2, 3, 4, 5, 6, 2 to the 6th power is 64. Is 64, which means I need to update my answer key. All right. 2 individuals have the genotype big A, big B, little b, big C, little c, and big D, little d. How many different types of gametes can be produced? We have 1, 2, 3, 4, 4 different alleles, heterozygous alleles, 2 to the 4th power equals 16. What are those gametes? Now this is a good opportunity to practice some Punnett squaring, but um, the only way to do this is to take 2 genes at a time, right? There, ye old Punnett square. And again, Trisha has a really great practice of color coding all of these. Um, I don't personally feel the need to do that, but if it helps you visualize where things come from, definitely do it. I will even do my speedrun version here and not even draw the box, but you know. At a certain point, the repetition becomes so automatic. Cool. So those are your gametes. Everything inside the box. I'm not going to rewrite these. Everything inside your Punnett square is a possible um, genotype for these gametes. And then of course you would combine all of those. So if you got your, yeah, you could make an even bigger Punnett square where you're crossing your AAs and your ABs, or excuse me, your ABs with your ABs with your ABs with your ABs, right? And your CDs with your CDs with your CDs and your CDs, right? and on it goes. So these are all of your possible combinations. It's not enough to just create the box and count up the number of squares and say there's that many individuals or that many possible combinations, because some of these combinations are going to overlap. Some of them are going to look the same. And I'm actually, no, in this particular case, they would not. They would not look the same. So each of these would represent a unique individual with a different genotype. Okay, you set up a Punnett square using all the possible gametes for both individuals. How many offspring squares are in this Punnett square? So if you used 16 gametes across the top of the Punnett square and 16 down the side, 16 times 16 gives you 256. You really can use this two-dimensional visual representation and solve it as an area of a rectangle problem, um, where each box is just one unit, right? 16 times 16 equals 256 offspring possibilities. Right. If you completed this Punnett square, how easy would it be to find all of the offspring squares that contain the genotype big A, big B, little c, big D, little d? The answer is it would be very hard. And it would be very easy to make a mistake. So we're not going to do it that way. We don't recommend that. So given that the genes are all on separate pairs of, you certainly can do it that way. I'm going to back up a little bit. You certainly can. However, I can't recommend it because it would be very tedious and you are very likely to make a mistake. Question E, given that the genes are all on separate pairs of homologous chromosomes, what other methods could you use to determine the probability of these individuals having any offspring with this specific genotype? You could handle each gene pair as a separate cross. So you just do these single crosses like we did up above, and calculate the probability that you get your big A, big B, little c, and big D, little d, and then multiply those across, right? So actually we can do that. We can do that. So big A is 50%. We've got one here and one here out of four. That's 50%. Big B, little b, big B, little b, big B, little b. Again, we've got 50%. Little c is only 1 in 4. It's only 1 in 4. And we had big D, little d. We've got 2 of those. It's 1/2. So to find out our probability of big A, big B, little c, big D, little d, we simply multiply 1/2 times 1/2 times 1/4 times 1/2 equals 2 is 4, 4 times 4 is 16, 16 times 2 is 32. Yes, 1 in 32. 1 in 32. And that is our last problem with 5 minutes to spare. Took me a little bit to warm up. Fortunately, my voice seems to be doing just fine, which is great. Get 8 hours of sleep a night if you can, y'all, for as many years of your life as you can. I highly recommend it. Don't be like me. Okay, that is our worksheet. Good luck on your exams. Review all of those things that I put a star on. They're very likely to show up. I think there was another one. Yeah. Spend some time with Punnett squares. Spend some time with this flow. Will absolutely help. Um, do not forget to turn in your worksheet for a completion grade, not for correctness, for completion, for those sweet extra credit points. And I will see you all next week.